Good morning. My name is Albertina Lee, and I am a research intern of the Lingappan Lab, part of the Children's Hospital of Philadelphia and Penn Medicine. Today, I'll be talking about my project on the implications of neonatal hyperoxia on prematurity-related cardiac disease in the atria and ventricles. Our lab is interested in decreasing morbidities in preterm neonates, where our long-term goal is to devise new therapies or drugs to improve the morbidities in this population. Preterm birth is associated with a variety of complications. There are factors both in the antenatal and postnatal periods that affect the development of different organ systems in the preterm babies. Among these is the developing heart. Preterm cardiovascular morbidity is multifactorial, where adverse factors in the postnatal period, such as sepsis and malnutrition, can also affect the developing heart. Therefore, preterm birth effects on development increases risk of long-term cardiovascular issues in babies born preterm. In this study, we specifically want to determine the effects of preterm birth on the heart in regards to characteristics of high oxygen supplementation. While preterm babies require oxygen supplementation to survive, exposure to high concentrations of oxygen predisposes to the development of many morbidities associated with preterm birth. Therefore, we aim to understand the chamber-specific effects of hyperoxic injury in the neonatal heart. Our goal is to identify mechanisms related to outside recovery and repair mechanisms following injury, and we hypothesize that neonatal hyperoxia exposure will lead to increased fibrosis and decreased microvessel density. We capture the effects of hyperoxia in the heart using histological and transcriptional assays. To determine the effects of postnatal hyperoxia on a human heart, we've analyzed the morphological and transcriptomic effects in the hearts of a mouse model that has matured to 2 months of age. The specifics of this mouse model featured two groups of mice pups. The experimental hyperoxia group was exposed to 95% oxygen between postnatal days 1 to 5. On the other hand, the control group mice were exposed to room air. Both groups were allowed to recover and mature to 2 months of age before their hearts were harvested. The hearts from the two groups were then stained for histological analysis and then processed for RNA sequencing.